Good morning. My name is Huda Zogbi, and it's my great pleasure to speak with you today about some of the work we're doing on Rett syndrome. I really regret that I couldn't be with you in person. Unfortunately, I had a conflict which kept me from overseas travel. But I'm very, very grateful to the organizers of the meeting and to Olga for giving me the opportunity to speak with you via videotape. As many of you know, my journey with Rett syndrome has been a long one. It has been from 1983 when I encountered my very first patient with this disorder. And we're fortunate to be able to have found a gene that causes Rett syndrome. And since then, we've been trying to figure out exactly what happened when this gene, which we call MACP2 for short, is mutated. What exactly happened inside the cell? What happens to the brain cells when they communicate with each other? What kind of cells cause the behavior that we see in children with Rett syndrome? And what can we do about all this? So today, in this very brief video, what I'd like to do is give you a very brief update about the work that's ongoing in our lab. Specifically, I'm going to give you an update about some of the more recent things, many of which are unpublished or some are recently published, just for you to get a feeling about how we're approaching this problem and what we hope to do so that we can help individuals with Rett syndrome. As I mentioned to you, this disorder is caused by mutations in the gene we named MACP2. And what we do know is that these mutations inactivate the gene, which means the protein loses its function when we have these mutations. So Rett syndrome is caused due to loss of function of this protein. When a protein loses its function, one way to understand the disease is to try to know what is the exact function of that protein in the cell. And we know that this is a protein that sits inside the nucleus of many, many cells, but it's very abundant in the neurons. And we know that it's critical for us to have very normal level of various genes that regulate our brain cells' function. Exactly how the protein does it is a little bit challenging, and we and others are trying to figure that out. We know that it sits on the DNA to regulate the expression of many genes, but exactly what happens after it sits on the DNA to do this regulation is what's being intensively worked on. And what we also know is that the levels of this protein really matter. We have to have it just right in the brain cells. If we have too little of it, we're going to have Rett syndrome. If we have too much of it, we're going to have what's known as the B2 Duplication Syndrome. So one of the strategies we've taken is to try to perhaps figure a way to boost the level of this protein. We do know that Rett patients, in half of their cells, they have a normal amount of the protein, and in the other half, they either totally lose the protein, or for many of them, they have a mutation that lowers the level of the protein. So one thing we're trying to do is to identify regulators of MACP2. Particularly, we hope for Rett syndrome to find regulators that will slightly increase the level of the protein in cells. Because if we can find those regulators, and if we can find maybe a small molecule, like a pharmaceutical, that might modify the activity of these regulators, we might be able to boost the level of this protein. This is ongoing work that's currently funded by the Rett Syndrome Research Trust, and while we don't have very specific regulators that boost the level of the protein, I'm happy to say we're beginning to find regulators that lower the protein, which can help the MACP2 duplication syndrome, and that gives me hope that it's a matter of time we can find additional regulator that boost the level of the protein. And this is one of the ongoing work that I imagine will take about a year to two to complete before we have the full picture of all the potential proteins in our body that might either increase or decrease MACP2, that might co-opt into modulating so that we can have ways to either boost the level of the protein in the case of some cases of Rett syndrome, or decrease the level of the protein in the case of the duplication syndrome. So this is an area of intense research in my lab, where I have a member of 3 to 4 people working on this area, because we feel this is really important and might be our path to find something that we call a druggable target, something we could modulate. And I hope that I'll be able to maybe report on that in the near future, or when the work is done. Another area of work has been, when this protein is mutated, what happened to brain cells that lack this protein? And there we have done a lot of work, and we have learned quite a bit. We've learned that this protein is essential for every neuron in our brain, and that different neurons might have slightly different vulnerabilities to the loss of this protein, and that different neurons might contribute to different clinical features of the disease. We've learned, for example, that inhibitory neurons are very important for the stereotyped behaviors. They are very important for the learning and memory we've seen, and they cause a variety of other problems, including susceptibility to seizures and so on and so forth. On the other hand, we're learning that the excitatory neurons are more important for symptoms like anxiety or tremors, and that both types of neurons are important for normal breathing. Probably breathing is so essential that it requires on all types of neurons. Now, why is studying the function of this protein in different neurons important? It's important because different neurons make different neurotransmitters, and they have different activities. By understanding that, and by understanding what symptoms are derived from which type of neuron, might help us sometimes design specific therapies for some of the symptoms of Rett. Again, this is work that's ongoing, and based on this work, we have been trying different pharmaceuticals to see what would happen if we try to boost the activity of the inhibitory cells. This is work funded by the International Rett Syndrome Foundation, and we're continuing with this work. It may be challenging. It may be that we might discover that we really have to boost the activity of all neurons to really overcome the symptoms of Rett syndrome. But we're trying that, and we're looking at that one step at a time. Now, neurons work in a network, and network is what determines behavior. And this is the last and most recent effort in our lab that I would like to share with you, and that gives me hope that we can reverse some of the symptoms of Rett syndrome, perhaps by modulating the neural network. So what do I mean by network? What I mean by network is that brain cells that work together and form a bundle of fibers to talk to another brain region, and through that activity, they can control behavior. We all know that the hippocampus is a very important part of the brain that's critical for learning and memory. Unfortunately, it's the part that degenerates in Alzheimer's disease, and that's why people become forgetful. And it's a part that we all need so that you can understand this lecture, so that we can form new memories every minute of the day. What we have learned from other studies is that there are some bundles or fibers that go to that part of the brain, and that if one stimulates those fibers in a brain part called the fornix, one can enhance the activity of the hippocampal neurons. And indeed, there are clinical trials in humans that are ongoing now to enhance the activity of these bundles in hope of improving the function of the hippocampus, and in hope in improving learning and memory in individuals with Alzheimer's disease. So this inspired us to consider the idea that perhaps in the mouse model of Rett syndrome, where we know that the hippocampus is not working perfectly, and when we know there are deficiencies in learning and memories in both individuals with Rett syndrome as well as in the Rett syndrome mouse model, we asked what will happen if we apply deep brain stimulation to this bundle, which is called the fornix. Would that improve the hippocampal function? Now, some of you may have heard of deep brain stimulation in the context of Parkinson's disease. As some of you may know, there are over 200,000 people who've been treated with deep brain stimulation for a totally different area of the brain to affect the motor symptoms of Parkinson's disease. Here we are co-opting the same approach that's used in human patients, but we're using it in a Rett syndrome mouse model to see if that would help us improve the learning and memory. So we adopted this technology to the mouse model of Rett syndrome, and we were very pleasantly surprised to find that upon deep brain stimulation of the fornix, just for an hour a day for a period of a couple of weeks, when we wait about 3 weeks and teach the animals tasks that required learning and memory, we find that the animals learned much, much better. And we tested them on many tasks that involve hippocampal learning and found that those tasks all have improved. Importantly, we also found that the adaptability of the neurons, or we call it plasticity, which means the ability of the neuron to adapt and to accept new information and store information, also improved. In fact, we found that the plasticity of the hippocampus, as well as its function as measured by these learning and memory behavioral tests, improved almost to normal level. So this really gave us hope for a few things. It gave us hope that if we focused on one symptom of Rett and we knew where to stimulate, we could potentially stimulate that area and maybe improve some clinical features. It also told us that the Rett syndrome brain is responsive to deep brain stimulation. Now, this was recently published a few months ago in November in the journal Nature, and I should mention this work was supported by the Rett Syndrome Research Trust and by the Keck Foundation, and of course my funder, the Howard Hughes Medical Institute. But now we need to continue this work to really see if this can move into the clinical realm. What do I mean by that? I mentioned to you that we gave this stimulation for 2 weeks and we saw a benefit 3 weeks later. The next question is, how often do we have to stimulate? And how long does the benefit last? If we stimulate for 2 weeks and 3 weeks later we get benefit, if we test the animals 6 weeks later, would they still have benefit? And if they do, would that last till 9 weeks later or 12 weeks later? We don't know the answer of that. My colleague, Jianrong Tang, who actually did this work in his lab, has now designed experiment, and we're collaborating with him to specifically answer this question. We want to know how long-lasting is the effect of deep brain stimulation, so that we can figure out how often we must stimulate so that the brain is always plastic and is always adaptable. So this is some of the work that's currently ongoing. Once we learn how long-lasting is the effect and the frequency that we need for the treatment, we need to answer 2 additional questions. We need to answer if we do this now chronically, let's say we discover we have to do it once a month or once every 2 months. We don't know yet. We have to answer that. But let's say we discover the answer. If we start doing it as such every month, for example, would we continue to have benefit? Would that benefit persist into 1 year or 2 years? This is another question we must answer. And thirdly, we need to know if we start this treatment at a much later stage in the life of the Rett syndrome animal model, would we still get benefit? As you know, or some of you might know, we started this treatment in animals that are young adult or adolescents, if you will. So now what we wish to try is also test this in animals that are 1 year old and see would we still get benefit from deep brain stimulation. So this is one example of an area of research which we really took not knowing if it's going to take us anywhere. It showed us an interesting benefit, and it opened up a way for us to begin exploring therapeutic approaches that might one day be actually translated into the clinic. Because as I mentioned to you, deep brain stimulation is being done in humans, and particularly of this particular area of the brain is being tested in humans with Alzheimer. So we hope it wouldn't be too challenging to move that down the line once we establish its safety, once we establish how frequently it has to be done. That's something we can use in a human. Another thing we need to consider when we're doing deep brain stimulation is how are we going to test for it being effective? In the animal, we give the stimulation and then we train them, and then we see if they learn. We need to be developing clinical approaches where we can see what can we train Rett syndrome girls on to see if now they are learning better. So really this work has to be coupled with clinical work. And a colleague of mine, Dr. Bernard Suter here at Baylor College of Medicine and Texas Children's Hospital, is doing this work as we speak. He's trying to determine exactly how we're going to really measure that. And in addition to our focus on the learning and memory, we need to be thinking beyond just the hippocampus. As many of you know, motor problems are quite prevalent in Rett syndrome, and these girls have a lot of motor abnormalities, from the freezing when they try to walk, to differences in their ability to move swiftly, to balance issues, and so on and so forth. So now that we know that the Rett brain is receptive to deep brain stimulation, we would very much like to begin to test what other part of the brain network might we stimulate, and would that help motor problems? This is something we're just beginning to explore. The hippocampus was an easier access for us because we knew exactly what bundle of fibers to stimulate so that we can help enhance its activity. Now we need to do a lot more studies to really figure out exactly which part of the brain might be worthy of stimulation to improve motor output. So these are some of the examples of the work that's ongoing, and we hope that through this work, either through discovering maybe a way to develop a pharmaceutical to change the level of MACP2, either by or finding ways to maybe enhance the activity of specific subtypes of neurons, or manipulating the networks. We hope one of these strategies would help us bring some treatment and some relief to individuals with Rett syndrome. I hope these brief vignettes I told you about the work that's going on in our lab have helped you understand at least some of the strategies we're using, and I hope that at the time I go to Vienna for the 50th celebration of the first publication by the late Professor Andreas Rett, I will have some exciting updates to share. I'm very grateful to all the families who've participated in our research. I'm very grateful for all the funders that I mentioned for their support, including the NIH and my institute. And I'm also very grateful to all my colleagues that contributed to their studies. And I thank you very much for your attention.